yeast, flies, worms, mice. What are in any model organisms that people work with? What to this to date have been some of the most dramatic life extension results? Like, is anyone able to tweak one of these things and improve lifespan by 10%, or are people doubling the lifespan of worms and yeast and things like this?
Well, so I think the most dramatic one is a worm lifespan, where this age one mutant that Tom Johnson discovered was a partial loss of function. And it was thought that if you knocked out the gene completely, it was lethal, or that the worms couldn't develop from their juvenile stage. And it was shown a couple of years ago, well, probably 5 or 10 years ago now, that if you take those complete loss of function worms and wait long enough, they do develop into adults, and they live 10 times longer than normal.
Wow.
So you live almost half a year.
So what was that gene doing?
It's in the insulin signaling pathway. And so that's a general theme, especially in model organisms, that disrupting insulin signaling seems to extend lifespan.
Interesting.
And that, in terms of mammals, that tends to carry over. So the insulin and IGF-1, which insulin like growth factor 1 pathways, overlap a lot. They can activate each other's receptors at a certain level, and a lot of the downstream signaling components are the same. And so in mice and in humans, it looks like there's more evidence probably that IGF-1 pathway disruption is maybe beneficial for longevity. But in lower organisms, insulin signaling in general seems to be.
I see. And could you just give us a sort of insulin 101 here? What is the basic biology of insulin? What is its normal physiological role? And how does, from there, we can probably build on some of it further.
Right. I mean, so insulin's the primary hormone regulating glucose metabolism, right? So if your glucose levels go up, you have a corresponding rise in insulin, and that sort of instructs cells to start taking up glucose and restores normal blood sugar levels. And so diabetics, either have become terribly resistant to insulin, so it's not functioning well enough and their blood sugar gets out of control, or type 1 diabetics can't produce the insulin in the first place, and so have the same effect. IGF-1 is in the growth hormone signaling axis. And so growth hormone causes the liver to release IGF-1, and that tends to have an anabolic effect overall. But inside this, so like I said, both of them kind of interact on the same receptors, and inside the cell, they tend to activate the same signaling cascades. And so it's still kind of a mystery in biology of how these things are partitioned. Like why does it seem like these two hormones can have slightly different effects through the same receptors in some cases, or similar looking receptors, and then hitting the same downstream pathways? Interesting.
So, so I mean, insulin's obviously involved in diabetes and other metabolic stuff. I've also heard more in different ways about this thing called mTOR, and I know that's really important for metabolism. But how does that start to tie into this?
Right. So that is one of the direct downstream effectors of both of those pathways. Right. So there's a direct sort of kinase cascade that goes through a bunch of steps, but mTOR is directly downstream. So when you stimulate insulin, you'll activate mTOR or IGF-1. And so that whole axis, I think, is probably thematically the area where there's been the most sort of success and consistency in regulating lifespan, is this growth hormone to IGF-1 to mTOR axis, which line up a lot of the interventions in.
I see. Yeah. So these things are sort of all tied together in some of these key molecular pathways that have to do with metabolism, and obviously metabolism and aging are going to kind of go hand in hand in many ways. mTOR, right? I'm not going to remember the full name, but it's something like target of rapamycin, this other drug.
Mechanistic target of rapamycin. So it's changed names a few times because it was violating some naming conventions, but that's the current standard.
I see. And what is rapamycin, this drug that can affect mTOR? Because I know that there's been some interesting results to do with rapamycin fairly recently.
Yeah, so rapamycin's been the drug that's been the most consistent for lifespan extension, and it is an inhibitor of it. So mTOR is actually in two different complexes. So the history of it was that rapamycin was known first, and it was actually discovered as an antifungal, but then realized to sort of suppress growth in all kinds of cells. And through, the pathway just wasn't known for a long time. And a guy named Michael Hall discovered in yeast what the target was and named it target of rapamycin. And then David Sabatini and others discovered the mammalian version and named it originally mammalian target of rapamycin, which is how it became mTOR. But because of the naming conventions, it was changed to mechanistic target of rapamycin later on. And afterwards, it was discovered that there's another protein complex that uses the same catalytic subunit. So mTOR is actually in two different complexes that have different functions. mTOR complex 1 is the one that's sensitive to rapamycin that people had been studying for a while. And mTOR complex 2 it was named for being insensitive to rapamycin. There's a component of it that actually has insensitive in its name. But it turns out mTOR complex 2 also gets inhibited when you treat chronically with rapamycin. And so actually both of them are probably playing a role in the effects of rapamycin when you treat long-term.
So when you activate this mTOR pathway versus when you suppress it, like high level, what are the overall effects? Is one of them sort of growth, promoting metabolism, promoting,
exactly.
and one of them, gotcha.
Yeah, mTOR is kind of the decision point in the cell. It's a node that decides whether the conditions are right for growth. And so missing a lot of different things. If you're missing amino acids or you're missing growth factor signaling or glucose is low, lots of those things you can dominantly cut off mTOR signaling and shut it down. You have to have all of them on to simultaneously to get mTOR to turn on and to start activating synthesis of proteins and amino acids and lipids. It's everything you need for growth, essentially.
I see. And so the very simple way that you often learn to think about things like, I mean, when you think about something like cancer and cell growth, right? Cancer is just sort of runaway cell growth. When you think about growth hormone or anabolic processes that are building up tissue and using more metabolic energy to cause more growth, that tends to be associated with growth on the one hand, which can be good, but also, bad stuff like cancer and aging. And then, of course, I think people hear a lot about these days the effects of things like dietary restriction. So if you just simply consume lower calories, this seems, to my understanding, to have certain beneficial effects. How does, when we're thinking about rapamycin and mTOR and this sort of really key metabolic molecular machinery here, is that sort of a very simple but intuitive way to think about these things, that things that are generally going to be growth promoting will tend to have pro-aging consequences just because you're going to get more oxidative stress and stuff happening, and then the reverse being true if you sort of slow down those pathways?
So yeah, in a general correlative sense, that way of thinking, it tends to hold up. And so, for instance, when it was discovered that rapamycin extends lifespan and calorie restriction we know extends lifespan in model organisms, the thought was, okay, so calorie restriction shuts down mTOR signaling, and this is all one pathway. That hasn't really proven to be true as far as there's a few observations that contradict it. Things like if you just do gene expression profiles and things and just look at how the tissues are behaving under calorie restriction or rapamycin, several attempts to show that they were the same, ended up proving the opposite, that they just looked completely different. And there's observations like the fact that with rapamycin, you can start very late in life. So the first study that really showed rapamycin would extend lifespan in mice started at about 20 months of age. And you get nearly the full effect on lifespan extension starting at that age, where with calorie restriction, the earlier you start, the better it works. And by 20 months, it's just about too late to get any lifespan extension from calorie restriction.
I see. So in rodents, calorie restriction, the earlier the better. The earlier that you restrict the calories the mice are ingesting, the longer they're going to live. And then at some point, it becomes too late. But you're saying with rapamycin, no matter when you start giving the mice rapamycin, you get this increase in longevity. What kind of increase were you talking about here?
So generally, it's been around 15% for most of the studies in rapamycin. If you look at the ones where they've optimized the doses and it tends to have a better effect in females, you can get up around 40% with sort of the maximum tolerated dose and in the right gender. So
15 to 40% of the lifespan. But if one could somehow translate that kind of effect to humans, you're talking about decades.
Yeah.
Interesting. And so what else do we know about rapamycin? I vaguely remember learning that it's also immunosuppressant. Does it have any other uses, like clinically, for other things?
So the two things that it's been used for, one is an immunosuppressant, and it's still used sometimes for, particularly for transplants to prevent rejection. It's also been used as an anti-cancer drug, and in most cases, the tumors have developed resistance pretty quickly, and so it's kind of been abandoned as the front line for a lot of tumor types. But there is a disease called tuberous sclerosis, where the primary defect is hyperactivation of the mTOR pathway, and that leads to lots of little tumors. And so in that case, it seems pretty effective to target mTOR directly. And so it's kind of in rare tumor types, still being used as an anti-cancer drug.